All right, so now a presentation about microbes and how they drive soil organic matter accumulation in organic cropping systems. Welcome to the stage.
Thanks a lot. It's great to be here today. I'm Stuart Grandy. I'm from the University of New Hampshire. Before that, I'm an associate professor there. Before that, I was a professor at Michigan State, and before that, I was a postdoc at the University of Colorado. We do a lot of work on sustainable agroecosystems at the University of New Hampshire, including quite a bit of work in organic cropping systems. I've really enjoyed this meeting today. I've had some really fascinating conversations with other scientists and people working in processing industries and growers and certifiers and a real diversity. It's been really, really fun. So thanks to all of you for making this an enjoyable meeting. I think we would all agree that organic cropping systems are highly, highly dependent upon soils. We don't have the luxury of replacing the services provided by soils with chemical inputs in organic cropping systems. So we're dependent on things like decomposition and the generation of nutrients. We're dependent upon good soil structure for water delivery. And yet we also know that soils are incredibly complex. And personally, I find it a bit frustrating in that I feel like we're still throwing around a lot of assumptions and generalities about soils and soil biology and how they work, and that's not to place blame anywhere. It's based in the fact that soils are incredibly complex, and with new tools coming online, we're just finding out that they're more and more complex. But just to throw out a few of the assumptions that you often hear that I think have been very strongly challenged and are probably just simply not true. One is that fungi are better than bacteria. You hear this a lot, that you want a soil that's dominated by a fungal decomposition pathway. I think there's ample evidence that that's just simply a generality that doesn't hold. Yes, maybe in some cases you want more fungi than bacteria, but they're both serving very important purposes in the soil. Another would be related to soil microbial diversity. Soil microbial diversity increases with improved soil management, like the incorporation of more residues or compost and manure, or changes in tillage practices. And yet there have been multiple studies looking along management intensity gradients, and they often don't see increases in diversity with reductions in management. Soil communities are really, really diverse. Tens of thousands of species in a single gram of soil, billions of individual cells. There's lots and lots of diversity there. And so I think we need to look hard at these kinds of assumptions. Or even that soil microbial diversity is good. I mean, clearly we need diverse food webs and food channels and multiple trophic levels, and we need a highly functioning microbial community. It doesn't necessarily mean it's the most diverse. Again, microbial communities and soils have an ample amount of diversity in them. I would even argue that some of the simplest assumptions that we hold about the relationships between plant inputs and soil carbon accumulation are being challenged. We historically thought that humification processes involving lignin and lignin derivatives led to the formation of soil organic matter, and yet now we seem to believe that that's not really the case, that most soil scientists, or at least my peers, won't even use the word humification because they don't believe it exists. They don't believe that lignin derivatives are an important component of soil organic matter formation, and this raises all kinds of questions about how we build soil organic matter. And these are that particular issue, the role of microbes, the role of the quality of litter inputs, are things that I'm going to be talking about today, and hopefully, you know, I'll be helpful in answering one little question about the soil. But I think in general, I just want to say that I think we need to be really working very, very hard for science-based information about how our soils are working in organic cropping systems, because the generalities are just not going to get us there. So before I begin the formal part of my presentation, I'd like to start by thanking my awesome graduate student, Cynthia Kallenbach, who has been funded by the National Science Foundation and the USDA to do this work that I'm going to be talking about today. Okay, so this is the conventional model of soil organic matter formation, that plant residues enter the soil, they undergo some kind of decomposition process. There's only partial decomposition of lignin, so you end up with a lot of lignin-type compounds and derivatives that make up the majority of soil organic matter, right? And based on this model, you would expect that those kinds of residues that are recalcitrant, that have a lot of lignin in them, would be ones that contribute the most to soil organic matter. Ones that decompose very quickly, like clover residues that promote microbial activity, would reduce soil organic matter. And yet, the new models of soil organic matter formation, are arguing that, in fact, lignin doesn't stick around in soils, that it decomposes very quickly. And we've discovered this using a number of different techniques. I mean, we've very much evidence now to support this, using isotopes, using very advanced spectrometry. We know that lignin is not sticking around. Sure, we know from litter bag decomposition studies that it decomposes a little more slowly than carbohydrates, but it's still it is not persistent in soils. Microbes have been evolving for a very long time. They have the capacity to break down lignin in relatively short periods of time, so virtually all organic matter is going through the microbial biomass pool. So what is it that becomes organic matter? It's actually the microbes themselves. It's the bodies of the microbes, or the necromass, the microbial cells, wall fragments, the intracellular components, the cytoplasm. These are the things that are becoming organic matter. They're not inherently chemically resistant to decomposition, okay? It's not that they contain complex chemical chains or ring structures or anything like this. It's that they stick to minerals, and it's that physical protection, that association between the clay and the residues that protects the microbial fragments and turns them into long-term stable soil organic matter. And again, the evidence for this is quite strong. We've got isotope ratio data, spectrometry data, and so on. And when we look at this, what becomes really important now in determining how much organic matter we're going to build in a system is not the amount of lignin going into the soil. It is all about the function of the microbial community. It has to do with its microbial growth efficiency. So how much of that carbon a microbe takes in is converted to CO2 versus incorporated into biomass. That's going to make a huge difference in terms of how much carbon becomes soil organic matter, okay? Things like the microbial growth rate. How fast is the community growing? How big is the community? Even things like the cellular structure of the microbial biomass is going to matter. We've studied plant chemistry for a century, and we've done almost nothing to learn about the microbial chemistry. And it's actually the microbial chemistry that's contributing to soil organic matter directly. Sure, the ultimate input of carbon to the soil is through photosynthesis and plant inputs, but what's becoming soil organic matter is largely microbial necromass. And we know very little about the chemistry of that necromass that's becoming soil organic matter. We know very little, almost nothing, about the growth efficiencies of microbes. How much carbon do they take in and turn to biomass versus release as CO2? We know very little about the growth rates and growth efficiencies. So this is, to summarize some of these new concepts in soil organic matter formation, just a conceptual model. There's a lot going on here. If you have trouble following this diagram, don't worry about it. I think I've made the main point. But basically this shows on the y-axis the relative abundance of different chemical classes. And then you've got a continuum in the soil from litter to sand to silt to clay. And it's going from biochemically protected organic matter, so that would be lignin, so it's protected by chemistry, through physical and chemical protection. And then you've got plant-derived versus microbially derived carbon. And so basically the take-home message here is that when you move along this continuum in the soil from litter to sand to silt to clay, you're going from increasingly plant-derived carbon to increasingly microbially derived carbon. And we're also looking along an age gradient. The stable carbon is in that clay pool. And you can see, because it's all hashed lines, it's not the black plant-derived carbon, that it's all microbially derived carbon. And so this is arguing this conceptual model that old soil organic matter is derived from microbial biomass, and it's not consisting primarily of aromatic compounds derived from lignin. Instead, it's simple compounds, carbohydrates, nitrogen-bearing compounds such as amino acids, proteins, and lipids. Okay, so it's not lignin, it's those products of microbial biomass that stick to soil particles that become clay-associated, long-term, stable organic matter. And we have multiple kinds of evidence, again, isotopic evidence, spectrometry, and so on to provide the data, the backbone for this graph. Okay, now again, the argument for why we thought lignin contributed to organic matter for so long is a topic for another day. If anyone is curious about that afterwards, how we went down that path for so long, I'm happy to talk about it. So, given the importance of microbes in soil organic matter and microbial biomass, we set out to do a field experiment in an organic cropping system to test this idea that growth efficiencies, growth rates, biomass production are ultimately the drivers of soil organic matter formation. Okay? We also set about to test an idea that those residue inputs or management practices that promote growth efficiency and growth rates will be the ones that increase soil organic matter contents. And so we did this at a site at the Kellogg Biological Station, the conventional corn, soy, wheat rotation, and an organic corn, rye, soy, wheat, clover rotation. I'll stay right up front. This is not an organic system that's managed for maximizing productivity. This is a long-term ecological research site that has other objectives as well. For example, historically, there were not the kinds of organic matter inputs that you would expect to maintain maintain crop productivity in the organic system. But you can see that what we have used at this site is this clover cover crop. And that's an important part of the story here, I think, is this clover cover crop. And if you look, the biomass inputs are actually less in the organic system, the total biomass inputs, even with the clover cover crop. And the reason for this is that the total corn stover and wheat stover production is much lower because of the lack of fertility in the organic site. Okay? The tillage is higher in the organic site because it's tilled for cultivation, and yet the soil carbon is higher in the organic site. So we have a system with lower biomass inputs, less or more tillage, and yet higher carbon. That's very counterintuitive, right? We typically expect carbon inputs are associated with soil carbon. We also expect that increasing tillage in general reduces soil organic matter, especially in sandy type soils like this. This is a sandy loam. And yet we're seeing an increase in organic matter. So why is that? I want to point out before I go further that the clover cover crop is a really important component of this system. You can see here it's diversifying the inputs, it's changing the timing of inputs, and it's increasing the inputs of carbon that have a low C to N ratio that would be expected to promote high growth efficiencies and high growth rates. Microbial growth efficiency is tends to be higher on high quality materials. Okay. So the hypothesis here is that changes in microbial physiology and carbon allocation will increase microbial inputs to more stable soil organic matter fractions. So we've got this hypothesis that microbial biomass will become necromass and contribute to organomineral stable soil organic carbon. High quality resources and frequent carbon inputs in the organic systems were expected to increase growth efficiencies and increase microbial growth rates. And we expected that these changes would be in part help explain these differences in soil carbon despite the tillage and despite the differences in tillage and total biomass inputs. So again, this was a field experiment where we measured microbial growth efficiency and microbial growth rates at the field level across time. And these are just how we measured microbial growth efficiency and growth rates. MGE is measured using C13 labeled glutamic acid, and MGR, or microbial growth rate, is measured using nucleic acid production as a proxy for MGR, and the soils are incubated with an 18O labeled water. I don't want to go into the details. Along with measuring MGE and MGR, microbial growth efficiency and growth rates, at the field scale, we carried out a second experiment where we took cores and put them in the soil, and then we added 2 different compounds to different cores. We either added 13C labeled glucose or 13C labeled a ring, I think it was syringol or oxalic acid. And we were interested in the fate of those new inputs in the organic and conventional systems, okay, and whether the higher quality carbon input, so the glucose, actually resulted in greater incorporation into stable soil organic matter than the ring structure. So again, we have 2 different levels of experimentation going on here. One at the field level, where we're measuring MGE, MGR, microbial biomass throughout 2 seasons, and then we have this core experiment, an experimental manipulation where we're tracking isotopically labeled substrates through microbial communities and into different soil organic matter pools. So we had a phenol core, that's the ring structure, and a glucose core. This is just a picture of the cores with the red clover cover crop in the organic system. And we traced the substrates into microbial biomass, phospholipid fatty acids, or the microbial community. So that was C13, PLFA, and into soil clay fractions. Interestingly, the microbial community structure determined using PLFA is quite similar in the organic and conventional systems. You can just look here at the relative size of the bars in each of these 2 sites. People have also used genomic approaches to look at the communities in these different sites, and they show similar kinds of results, that the organic and conventional sites actually have fairly similar microbial community structures in terms of who's actually there. But what I'm going to show you is that these communities function completely differently and have very different physiologies. And there's much greater biomass of the community in the organic site. That's what these middle bars are referring to, and the y-axis on the right shows the total phospholipid fatty acid. So again, a different method. This is using chloroform fumigation, showing much higher biomass at multiple time points in the organic system. And we also saw much higher microbial growth efficiency determined on glutamic acid in the organic systems. So in the organic systems, we estimate that 55% of carbon inputs are converted to microbial biomass. In the conventional system, it appears that 55% of inputs are converted to CO2. Now, if all carbon is ultimately going through the microbial biomass, growth efficiency becomes really, really important. And this difference of 10% iteratively over time could be very important. And we're also seeing greater microbial activity in the organic systems. So the turnover and growth of the microbial activity determined using O18 is higher. And as far as I know, these are the first studies that I'm aware of showing higher growth rates and growth efficiencies in organic production systems. We also are seeing from those C13 labeled cores, remember we added carbon that was labeled to cores and tracked it in the field. And I'm only going to show a small part of that because we have C13 PLFA data. We know which parts of the microbial community the carbon went into and things like that. But in general, we're seeing in both the glucose and phenol that the recovery of C13 in microbial biomass is higher in the organic than the conventional site. So more is getting incorporated into microbial biomass. I want to say that since this was done, we have more data points over time, and the phenol comes up a little bit. There's just a little bit of a lag time in the incorporation of the phenol into the microbial biomass, but 6 months later there was actually more phenol than glucose in the microbial biomass, and again it was higher in the organic than the conventional system. So as expected, yes, the ring structure does slow decomposition on the course over the course of weeks to months. But in terms of organic matter that's stable for thousands of years, hundreds to thousands of years, that doesn't really make much difference. But anyway, the take home here is that yes, we're getting more rapid incorporation in the organic system of C13 label into the biomass. And ultimately, this is the glucose recovery in different clay fractions, and we're seeing much, much higher recovery of C13 labeled glucose in clay associated with these macro aggregates. So what appears to be happening here is that in the organic system there's more rapid uptake of C13 label, and the turnover of that biomass results in the deposition of that C13 label inside aggregates on clays where it's probably quite stable. So just to synthesize, we've got these two systems. This is my last slide. We've got a conventional and organic system. The conventional system has more carbon inputs, less tillage, and yet it has lower soil carbon. One of the big differences is that the cover crop in the organic system results in higher quality inputs and a different timing of those inputs. We find that the microbial growth efficiency and growth rates are both higher in the organic systems. We think it's likely because of the input of the clover cover, because we know from culture work and a variety of different experiments that have been done in aquatic and culture systems that growth efficiency is positively related to the quality of carbon inputs. And then finally, when we're seeing more microbial residue inputs in the organic system than the conventional system, and this is building soil carbon over time. So we're seeing over time an increase in microbial residue inputs due to that increase in growth efficiency and growth rate, greater incorporation of new inputs into biomass. Those inputs get stabilized on clays, and we get an increase in stable soil carbon. I'm just going to end with this slide and say that I think going forward there are so many opportunities in organic cropping systems to try and find those processes that are underlying the biology that we need to maintain and increase productivity. And we have to go after the funding and do all kinds of different things to try and generate this kind of research that is really going to show the mechanisms that we can carry forward and apply across a wide variety of sites. And I also think that there are funding agencies willing to support this work. It's how you present it. As I said, my student has received funding from the National Science Foundation, the the USDA has been very enthusiastic about this work. I've received money from the Department of Energy for this work, and so we just have to go after that money, and hopefully in 10 years we'll keep making some progress. So thanks.
All right, questions?
Thank you very much. I've got two questions. One is that respiration is being promoted as kind of the soil biology test du jour, the Haney method, Salvita, et cetera. And so based on what you're saying, some people look at respiration as a measure of activity, which I'm not sure is the same as rate, but clearly it represents more CO2 being blown off. So first question is, what's your sense of respiration as a useful measure as it's being promoted for growers? And second question is, what about soils that are sandy and 5% clay, 3% clay? What's the implications of what you're finding?
Yeah. Well, respiration is not simply a measure of the activity because it encompasses both activity and growth efficiency. And so you could have soils with an equal size biomass, one's producing a lot more CO2 because the growth efficiency is lower. And that wouldn't be a great thing. In terms of broad brushstrokes, you could probably argue that soils with higher respiration rates are generally have a higher biomass. So I'm not willing to stand up here and say that we shouldn't be using respiration rate as a proxy for activity. I mean, I think until we come up with something better, it's reasonable. But hopefully we'll be able in the next few years to have more specific kinds of tests that are farmer friendly for measuring things that really matter. I mean, we've been doing some things with these kinds of tests looking at ratios of different kinds of functions and enzyme activities, and I haven't really gotten to the point yet where I feel entirely comfortable going to growers with them. But for example, we've been looking at hydrolase to oxidase enzyme activities, and across 10 sites now where we have management intensity gradients, agricultural management intensity gradients, we see consistent trends in those ratios of enzymes that are acquiring simple carbon versus complex carbon. So hopefully we continue to see improvement in that area. But yes, respiration is much more complicated than just the activity, but those tests have to be simple, and because of that, you have to be willing to sacrifice something. And I would still say it's a reasonable proxy for activity and biomass.
Stuart.
Oh, yes. Yeah, so that's a good question. In fact, we've done some, a colleague of mine at the National Center of Atmospheric Research in Boulder and I've developed a new model, a new carbon cycling model, a global carbon cycling model called MIMICS, the Microbial Mineral Carbon Stabilization Model. And we've modeled these carbon dynamics in clay soils and sand soils, and we do show in those sandier soils that more recalcitrant inputs, ones with a lot of lignin, should be expected to result over time in more carbon. But as soon as you start getting into a soil that's more loamy, that has silt and clay in it, that's where you would expect the higher quality residues to build organic matter because they're driving microbial biomass and necromass activity. But that hasn't been tested. But that's our supposition at this point. That's still in the very sandiest soils. You would expect the conventional ideas about soil organic matter formation to hold. But again, I mean, even at KBS, those soils are sandy loams, so they're fairly sandy, and we're seeing these dynamics where carbon accumulation is very closely related to growth efficiencies and growth rates and the incorporation of new isotopically labeled materials into biomass. So.
Stuart, thank you for that stimulating presentation. It's been very interesting. The old lignoprotein theory of humus formation had some explanative functions that were useful also for the practitioner. One of them was that young organic matter, young green manures, for example, have less of a residual effect in building organic matter than do an older green manure crop. Or manures that come from a cow, for example, which would have more lignoproteins in it, would have a stronger effect in residual building of organic matter than, say, organic matter coming out of the gut of a chicken. And that was useful, and I think also those observations from the practical side are based on reality. There's plenty of evidence that such things happen with respect to residue quality or organic matter quality. But if we think of organic matter quality in that sense, the younger material or the raw material has less effect in building soil organic matter. It's also that material which is more available to microorganisms, whereas a more stabilized organic manure would have a longer term, more residual effect in building soil organic matter. That's kind of standard organic farming. Could you explain to me, since your theory is so different in the sense, how organic matter pools are stimulated then by more stabilized organic matter sources to result in a longer term buildup of organic matter?
Well, again, I think that you have to consider the soil type that you're looking at, and I also think you have to consider the timing or the time scale over which you're looking. And I think that when we add recalcitrant residues or composts that have been processed by animals and therefore have more recalcitrant materials left over in them, that we're seeing an increase in carbon. I think those are largely visual observations, and I think that over time, over the course of several years, I think that you're more likely to see buildup of organic matter in sites that have more fresh residue incorporation that promotes microbial activity and aggregation and absorption of simple molecules on clay particles. And I mean, I don't mean to stand up here and discredit, you know, observations and the knowledge that farmers have of their own fields. I mean, I've got my in-laws are farmers. I worked on farms for years. I value and appreciate that perspective, but I just don't see the data for that, Walter. I mean, you can look across multiple studies, and there's the correlation between the quality of the chemistry of the inputs and the buildup of soil organic matter is extremely weak. And so I think the reason for that is that ultimately it's things like the microbial growth rates and growth efficiencies, the rate at which they're incorporating new materials into biomass are what's driving soil organic matter formation in soils that have any type of clay content. And that's going to be driven by the quality of inputs, but also things like climate and other management practices. So I guess I guess that's my perspective on this.
All right, other questions?
And I will say too that there's just really no evidence that there are these lignoprotein complexes forming in soils. And we can look and look and look using our most modern methods at stable soil organic matter, and we just don't see those lignoproteins that people hypothesize that we should see. The chemical extraction methods that were historically used to estimate different fractions of organic matter were entirely wrong. The proximate analysis technique that people used to estimate lignin often was extracting no lignin at all, none. And so we now go in and we say, oh, I'm using proximate analysis and this isn't a metric or a proxy for the amount of lignin. Well, we can now go in and look at the organic matter that's extracted using proximate analysis and see that there's no lignin in it at all. So this pool of organic matter that we were calling, that was 50% of SOM, that we thought was lignin based on chemical extraction. Well, there's no lignin in there at all. There's proteins, there's carbohydrates, and lipids. Those methods were entirely wrong. So I guess I don't believe in the lignoprotein formation at all, and I don't believe in humification being a process of soil organic matter formation. I think it's relevant in aquatic systems. I do think it's possible that you can have those kinds of browning reactions. You can do them in the lab. You can mix ring structures and nitrogen and see the formation of these lignoprotein complexes. You just don't see it in soils, and you don't see evidence of lignin derivatives accumulating in soils as these long chains that people historically thought.
All right, thank you so much.